Good afternoon, A&P students. How's everybody doing? Okay, I'm gonna do some Zoom reviews for our midterm test that starts next week, and I'm gonna actually do it off of the PDF files because they don't have the keys in them, and I'm gonna key them as I enter them as I go. So this would be excellent if you're not able to download the PowerPoint, which I strongly recommend because it has the answers in the notes section. Then you can actually watch this tape. I'm gonna go through the PowerPoint or the PDF file of the PowerPoint. Okay, so let's see where we're going here. So I'm going to start out and review a little bit on stages here. Okay, I'm going to share my screen with y'all. Hang with me. Hang on. Share screen. Let's see, here we go. Okay, you should be able to see that now. Okay, all right, let's see what we can do here. Let's walk through it. Okay, so oops, going to go pretty fast. So here we go. Anaphase here at both blues. Our stages of mitosis is a whitefish blastula. See if I can find a better way. I'm going to have to use that one rotation of the mouse, so bear with me. And then we've got a prophase at blue and metaphase at red, prophase at yellow, telophase here at blue, left blue, and then anaphase at the blue on the right side here. I've got the mouse on. Blue here is interphase, which is always kind of tricky, right? Interphase, you don't see any chromosomes. These stained for chromosomes that you can see here, and a metaphase there. Telophase and a metaphase, blue and red is metaphase. Then we have a prophase at blue, a anaphase at red. It's a whitefish blastula. And then here we have another one here with red metaphase, blue right prophase, and probably prophase here at blue. Could be a late telophase. I think it's prophase, but those two both look the same, so that could be something interesting going on there. Then we have telophase at red, interphase at yellow, and prophase at blue. Now we're looking at some onion root tips, right? And remember, I told y'all in class that whatever's most common is interphase. So yellow and all these that I'm touching now are all interphase. Blue is anaphase. Early prophase, see they're starting to coil up there. And of course, red is metaphase because they're lined up left to right. So metaphase red, anaphase blue, anaphase red, interphase yellow, prophase here. There's a big prophase at the blue there. Another, oh, look at that. That's telophase with that cell plate right there in the middle. That's a good one. Interphase yellow, anaphase here and here at blue, and then a late telophase there at red. Telophase at red, metaphase at blue, and interphase at yellow. Messed up looking prophase, anaphase yellow there, blue, anaphase, and there's another telophase at red. Okay, now we're going to start our looking at epithelium. And remember, this is the slide that we would normally do where we would scrape the cheek cells out, and they are stratified squamous in the mouth. But when you put them on a slide, they become simple squamous. You make a flat slide out of them. And here's your cheek cells at 100 power and at 400 power with bacteria and stuff. Now, if you remember, when we looked at this, we start out, we're going to look at simple squamous in the kidney. This is the kidney slide. To your right here, what we're looking at is the cortex. And over way to your left, that we kind of start to see the beginning of it right in here, is the medulla. Cortex means outer layer. Medulla means middle layer or outer layer. Outer layer cortex, lower or middle or inner layer is medulla. We're going to blow this up to 100, 400 power and look at the edge of this glomerulus. So I picked this slide just because this is a very unique place to see simple squamous. Your book, Hammer and textbook, use simple squamous in the alveoli of the lungs, but I like this. So kidney, 40, 100, and I see what I call meatball and spaghetti in here. These are the nephrons of the kidney with the glomerulus and Bowman's capsule around it, and these are the tubules of the kidney, and they're all simple cuboidal right through there. We're blowing this up, and there is simple squamous side view surrounding the glomerulus as it will probably appear on your midterm test. Here is some cuboidal of the tubules, but I will show it usually where you can't see the glomerulus. If you see the big meatball, then you're looking, I'm probably testing you on the simple squamous lining the edge of this capillary bed. This is the nephron of the glomerulus or the nephron of the kidney. I can make it silver looking by changing the color, still see flat pancake side view. Now we're going to the small intestine, and this is fascinating. And one of the reasons epithelium is so tricky is because you don't know what to draw on here. Just to give you a quick idea, the blue is smooth muscle. We're going to come back and look at this. And this is the small intestine, so this would be chitlins. And then we're going to go down these fingers in here called villi and blow them up and look for the simple columnar lining them. This is the lumen where the food would be in the intestine, and we have connective tissue up in here and glands up in here that make secretions that get put into this lumen. So we're going to go to 400 power, so 40, 100, 400. And here is our epithelium. Here's the basal membrane right here that I'm rubbing. And from here to here is simple columnar epithelium with goblet cells stuck in here and microvilli, the structure on the edge. So simple columnar epithelium, small intestine. It's up here too on each side of the villi from here to here. And you're seeing stacking, so you're seeing other cells in the same row that we're looking at. So it's like, if I'm looking at cells like this, I can see the ones behind them who are all on the basal membrane. Hope that gives you a little bit of help there. All right, next slide is I can stain them. This is a slide with goblet cells stained red. You can see microvilli and goblet cells here, or mucus actually pointing out there. And now we're going to go to stratified squamous. Now, in Ammerman's textbook, she used the vagina slides for the non-keratinized, meaning not having a hard flaky layer on the outside. We're going to look at the, this is the esophagus of the rat, and then we will look at skin for keratinized stratified squamous. So here we go, at 40 power, we want to go to where the blue arrow is, to 100, and then 400 power. So from here to here, stratified squamous, esophagus. And here it is, flipped over. Looks like the landing on the moon or a lunar surface. I'm seeing 3D. You can see here is the stratified squamous, esophagus. Then we're going to go to the skin, and we want to get this outside layer of the epidermis of the skin for stratified squamous keratinized. And then we have this huge layer in here, the dermis, which is going to be dense, irregular connective tissue coming up in just a minute in the next PowerPoint. Here's that dead stratum corneum outside flaky layer, and that's 100 power, and come down to 400, and you can see the papillae, little loop-to-loops in here, and from here to here is the cells attached, and there's that dead stratum corneum layer. Stratified squamous keratinized skin. There again, I like it. I got several pictures of it. And now we're going to go back to that medulla, the kidney. Here's the medulla. This is the other end from this slide. This is the far right, and there's the far left. Going back and forth. Meatballs, again, simple squamous lining the glomerulus. Then the medulla has these cubes. You can see them from far off here. We're at 100 power. When we go to 400 power, we can see these lines. There we go. Glamour shot, simple cuboidal epithelium right through there. And these are the tubules, those tubes that where urine is concentrated and made. Simple cuboidal kidney. Simple cuboidal kidney. Again and again. Glamour shot there, arrow at simple cube. Then we can slide to the thyroid gland at 40 power. This is our thyroid gland. And we're going to bring it in from 40 to 100. Notice it looks like lakes, catfish lakes flying over the delta of the plain. The levees are all simple cuboidal, and the fluid in here are precursors to the hormone thyroxines T3 and T4. Bring it in, and you can see the levees of these reservoirs are simple cuboidal. This would be simple cuboidal thyroid. Okay, now we're going to another one, and this is another cool slide. This is the trachea. Notice hyaline cartilage here, fat here, glandular stuff here, loose connective in here, and we're going to blow up on this edge where the windpipe would be for pseudostratified columnar ciliated. Here we are at 100, 40. Actually, that's 40, and then probably 100 here, 400 power. Notice the cilia, notice a goblet cell over here. And this is the pseudostratified, there's the basal membrane, pseudostratified columnar ciliated, and here it is at all. And you can see the goblet cell makes mucus that gets these hairs sticky. When we breathe in on a dusty day, those particles are caught in that dust, and mucus and bubble together to make boogers, okay? So my booger epithelium here, pseudostratified columnar ciliated. I'm going to back up, and here it is at probably 400 power, and then this is at 1,000 power or all there. Sorry for the jumping around. Then we're going to go to our last one, transitional. And this is shot at actually the ureter. So I actually need to reshoot these and get the bladder slides, but I use ureter slides, 40, 100, smooth muscle around it. Looking in here, for this is our transitional, and at 400 power, here is transitional. Notice the fluffy, stratified, maybe cuboid, stratified, looks like stratified squamous or stratified marshmallow cells. Here's a slide here, and you can see these are pretty big, and it is a stratified layer, and it's stretchy. So it looks like this on an empty ureter, but when it gets stretched out, it can look almost like skin. So this is called transitional because it's got a stretchy epithelium that can stretch out and shrink back in. So it's a stretchy epithelium. All right, and that's my picture I think that I have for transitional on the midterm right there. Okay, and that's epithelium. So we're going to take a break, and I'm going to film another one here in a minute for connective tissue, and then we'll have one for muscle and nerve, and we'll have y'all 3 videos out there for y'all maybe later tonight. All right, y'all have a great day. Bye-bye.